Hi guys, today you're going to work on your BRCA lab. You obviously collected data yesterday on this lab, but you have no idea what the concept of BRCA actually is. So what you're going to do is you are going to start by reading through the intro to the lab. What is a cancer cell and how does BRCA actually relate to this type of situation? So read through the intro here. Here's the BRCA1 and the BRCA2 genes. They're found on chromosomes 13 and 17. You're going to read a little bit about what BRCA might mean, as well as the words oncogenes and tumor suppressor genes. So there's a think about it question here that goes into it. Do you believe oncogenes are good or bad? It's your opinion, so you're not wrong. Another opinion question in here. Would you expect a tumor cell to possess a tumor suppressor gene? That's an interesting question to bring up. And a prediction. Do you personally believe that the BRCA genes are good genes or are they bad genes? You're going to make a guess right here. And from that hypothesis, we're going to see if your data leads to the conclusion of BRCA genes being good or BRCA genes being bad. So yesterday, you guys ran all 4 of these lanes. Lane 1, you had the DNA ladder. 2, you had the BRCA gene. Lane 3 was that of a normal cell. So tissue from normal cell, and lane 4 was tissue from a tumor cell. So I know in this particular class period, we had a hard time when we ran our gels. A lot of you guys punctured through or didn't get very good steady data, which is fine. The main purpose of gel electrophoresis is to do what? How do we sort genes? What directions do they usually travel in and why? Make sure you're utilizing this picture that's right here for you, help you with those questions. Then you get to the lab analysis. Take your picture from yesterday's lab. Yes, I know that you might not have clear data. That's okay. Place your picture in this spot right here. Then what you're going to do is looking at both of these pictures, you're going to put a circle around things that might be similar between them and a square around things that might be different between your 2 gels. So some of you guys are going to have a lot of squares and not a lot of circles. So if we take a look at this lovely gel, you see the DNA ladder in lane 1. What this is representing here is the actual BRCA gene. So the BRCA gene is present in lane 2. That's our control. Lane 3 is samples from a patient that are normal, non-cancerous cells. And what we notice here is that normal cells contain the BRCA gene. Tumor cells that were run in lane 4 do not contain the BRCA gene. So from this information, you're going to answer questions 15 and 16. Do you believe, whether you use your lab gel or the stock, which is this one right here, do you believe that, BRCA should be found in all these other places? Is it in normal tissues in lane 3? Or is it found in tumor cells in lane 4? Okay, based off of that then, what can you conclude about the BRCA gene? Is it a tumor suppressor? Or is it an oncogene that would cause cancer? So you're going to explain your reasoning. Complete this. Once you've done this, then head back into your Google Classroom. When you head into Google Classroom, you can start working on corrections that you might have to your Shark Tank presentation, and that can get resubmitted to me. If you and your partner are choosing to not complete any corrections, that's fine. You'll also be able to work on the GMO research if you have to make any corrections to that particular PowerPoint as well. So you have many opportunities to get work done in this class, to do makeup work, but you must have the BRCA lab done and turned in by the end of the period.